Successful RNA purification using the Monarch Total RNA Miniprep Kit can easily be accomplished by following these helpful tips. Before your prep, it is important to work in an environment free from RNases. Always wear gloves, use RNase-free glass and plasticware, and clean your work areas. We recommend wiping your benchtop with a cleaning agent such as RNase Zap. To maintain RNA integrity of your sample prior to the prep, samples should be frozen at the time of harvest or protected by a dedicated reagent such as the Monarch DNA and RNA protection reagent, which is included in our kit. Cultured cell pellets or aliquots of blood preserved with this protection reagent should be mixed thoroughly by vortexing or pipetting prior to storage. Small pieces of tissue less than 20 milligrams may be stored directly in the protection reagent. However, larger tissue samples should be homogenized in protection reagent prior to storage. If samples have been preserved in the protection reagent, do not remove the liquid prior to processing, as the protection reagent also begins the lysis of the sample. It's important to avoid thawing your sample in order to preserve its integrity before extraction of the RNA. Frozen cultured cells can be thawed briefly before the addition of the RNA lysis buffer, but most other sample types should not be thawed in the absence of the protection reagent or the lysis buffer. These samples include tissues, blood, bacteria, yeast, and plants. Ensure that your samples are completely disrupted and homogenized in order to release all of the RNA and maximize your yield. Mechanical homogenization, for example, with a bead homogenizer, can boost your yields beyond those produced by proteinase K digestion alone when you're working with tissue. Mechanical homogenization is also recommended for tough-to-lyse samples such as plant, bacteria, and yeast. When using mechanical lysis and homogenization, multiple rounds of homogenization may be recommended. If you do use multiple rounds, place your sample on ice for approximately 1 minute in between rounds to prevent your sample from overheating. If you are working with tissues or leukocytes, it can help to preheat your heating block to 55 degrees Celsius before starting your prep, so that the heat block will be at the correct temperature when you get to the proteinase K incubation. Please be aware that when working with mammalian whole blood, the proteinase K incubation is carried out at room temperature. We also recommend preparing a master mix of DNase I and DNase I reaction buffer prior to beginning if you are performing multiple preps at once. Be sure to follow our guidance on the recommended sample input amounts in order to ensure that the buffer volumes are appropriate and that the columns are not overloaded. This is a common mistake that quickly can reduce yield, purity, and integrity of your RNA. After sample lysis, perform all steps at room temperature. This will prevent detergent from precipitating in the buffers. Do not place your samples on ice after lysis. Before applying your sample to the genomic DNA removal column, be sure that you label the collection tube, as you'll be discarding the column after you centrifuge. After putting your sample through the genomic DNA removal column, make sure that you save the flow-through. This flow-through contains your RNA. If you want to capture total RNA, including small RNAs, add 1 volume of ethanol to your flow-through from the gDNA removal column, in preparation for binding it to the RNA purification column. If you want to exclude RNAs smaller than 200 nucleotides, only add half the volume of ethanol to your flow-through from the gDNA removal column. It's very important to perform all wash steps in the protocol in order to produce highly pure RNA. Be sure to spin your column for 2 minutes after the last wash. For low-yield samples, including muscle or brain, or for low starting inputs, consider eluting the RNA with 50 microliters of nuclease-free water in order to get concentrated RNA. It's important to have a concentration above 20 nanograms per microliter if a microvolume spectrophotometer, such as a nanodrop, will be used to measure concentration and purity. In some cases, silica particles from the column matrix can be found in your eluate. To ensure that this doesn't affect your OD 260-230 ratio, you can centrifuge the eluate for 1 to 2 minutes at 16,000 times G and pipette the aliquot from the top of the liquid for measurement on a spectrophotometer. While nuclease-free water is provided in this kit for the elution of RNA, adding EDTA to a final concentration of 0.1 to 1 millimolar can protect RNA samples that will be stored for an extended period of time. Additionally, it's good practice to store the eluted RNA in aliquots to avoid excessive freeze-thaw cycles. We hope that these tips have been helpful. If you have any further questions, our tech support staff would be happy to help. Contact us at info@neb.com or online at neb.com/tech support.